We go with our notes for chapter 6, which is on kind of the basics of metabolism, talking about energy and the way energy can change forms and be transferred and how enzymes help that process out. So the sections that we're going to be looking at in this from this textbook, cells and the flow of energy. So talking about specifically in cells, metabolic reactions and how energy is transformed, looking at specific metabolic pathways and how enzymes help with those metabolic pathways, and then taking a look at the basics of an oxidation or reduction reaction and the roles they play in metabolism. So first off, just some definitions. Energy is the ability to do work or bring about a change. Okay, so ability to do work. And there's two different types of energy. Kinetic energy is the energy of motion. So think water going over a waterfall. And this is usually like mechanical energy, like doing the work. Versus potential energy is stored energy. So this might be the water at on the other side of the dam before it goes over the dam, or a stretched rubber band being ready to be shot, or a bicycle on the top of a hill. Or, in the case of biology, we're talking about chemical energy, right? The energy is stored in the food that we eat. And two different laws of thermodynamics are at play when we're talking about energetics in biological organisms. The first is the law of conservation of energy. This is one that you've probably heard of before, probably talked about it in your chemistry class when you're balancing chemical equations. This is basically the idea that energy cannot be created or destroyed, but it can be changed from one form to another. And so photosynthesis does this, right? It captures the energy from the sun and uses that energy to form carbohydrates from carbon dioxide and water. So it rearranges the carbon dioxide and water to make carbohydrates. But a lot of the energy gets lost as heat as well, and that's an important thing. We saw that back here too. We've got energy coming in. We're losing some of the energy as heat. The plants are losing some of that energy as heat. This guy is eating some chemical energy and losing some of that as heat, but using some of it as mechanical energy. So that's really important. We'll be talking about heat a lot later. The second law is the law of entropy. Entropy is sometimes talked about as like the amount of disorder. And so when energies change from one form to another, there's always a loss of energy that is available to do work. So no energy conversion is 100% efficient. And so the majority of this energy is lost as dissipated heat, right? So when you're doing something, you're also generating heat. And that's that unavailable work. That heat escapes to the atmosphere and it can no longer be captured to do something. So if we think of, this is like our potential energy, our chemical energy gets converted to physical energy to contract this muscle, and this is also going to generate heat. Again, that heat just goes away. So you get the mechanical, like this is what you want, and this is the inefficiency part, the heat that's lost. So these processes that are occurring in cells, they're all basically just energy transformations. Every process in the cell is going to be increasing the entropy in the universe. It's always, everything we do is always creating some form of unusable heat. So it's always like basically reducing the amount of energy that's available to do work. But this is luckily able to be replenished by the sun, so we don't have to worry about it too much. But for example, we've got glucose tends to break down into carbon dioxide and water over time, right? And we talk about glucose as being a more organized molecule, so therefore it's less stable than these guys that are simple, right? This guy is more is less stable, is more easily broken down into smaller products. So it's more organized, has more potential energy, right? Has energy to give, to be converted, but it's less stable. Versus these guys are less organized, more stable though. They can't. It's harder for those guys to be broken down a little bit more, but they have less potential energy. In the cell, if we're talking, right, if we've got a lot of hydrogen ions on one side of the cell membrane, it's more organized, right? There's less chaos here, more potential energy. As these guys move, we can harness that energy, but it's less stable versus this is a more stable configuration. Kind of confusing, I know. So organisms, producers, use energy to create organized structure, right? So they are creating structure when they make the carbohydrates. But then other organisms called called consumers eat these guys. They use their potential energy to drive their own process. So they depend on that constant supply of solar energy to keep that whole process going. All right, so on to talking a little bit more about within the cell, metabolism specifically. So metabolism, we've used this term a lot already, is a sum of all cellular chemical reactions in a cell. And so you've got the reactants that take part in the reaction, and the products are what you make at the end. Free energy is the amount of energy available to perform work. And we can talk about reactions as exergonic or endergonic. And exergonic, these products have less free energy than the reactants. Basically, they're releasing energy. You may have also heard the term exothermic, right? And same idea. That's just you're releasing temperature, which is also energy. So it's like a form of exergonic. So they can almost be used interchangeably. So exergonic, the energy is exiting, is how I think of it. And endergonic reactions, they have more free energy than reactants, so they require energy input. So you have to put energy into them. So these guys can happen spontaneously, right? These things that require energy, they don't just happen spontaneously, right? You've got to put energy in for those to work. That energy, as we've been talking about, is in the form of adenosine triphosphate, or ATP, and this is this high energy compound. The ATP isn't really stored by the cells, it's just constantly being cycled between ATP and ADP, with the D being just diphosphate. And we've talked about the structure. Adenosine and ribose together make adenosine, and then you've got 3 phosphate groups. And you see here, this guy's got this wavy bond. This is like the high energy bond. It's easier to break that bond and get your ADP plus a phosphate. And so coupled reactions release energy released by this reaction can get captured in ATP, and then we can use that ATP to drive something that requires energy. So some things can release it, and we harness that into ATP, then we use that ATP and release that energy from there to drive something that needs an energy input to happen. So the ATP cycle, how we make it, so we've got our ATP, we can crack off one of those P's, and this is going to release stored energy. So that's going to, just release the energy. So then you're left off with an ADP plus a phosphate, just kind of hanging out. And so that's got to get recycled so that the cell can use it again. So now we have to have, we can make that back into ATP, but this is going to require the input of energy from other sources. So this is where cellular respiration comes in. We have to have the energy from the glucose to come in to fuel the process of turning ADP back into ATP so that we can send the ATP out to the cells to do the work. Coupled reactions, these are examples of reactions that require this ATP, and a great example for looking at this is the muscle contractions. The way muscle contractions work, we call this a sliding filament theory of contraction, and your muscle fibers have these 2 different proteins, actin and myosin, and the myosin binds to the actin and like pulls it along. And so here we have the resting shape, and this is combined with ATP, and it's got this form. The myosin has this kind of form where it's not noticed as not making any contact with the actin. We split that ATP into ADP, and that causes the myosin to change shape, right? So we're releasing some energy and allowing that to change shape, and then it's able to connect to that actin filament. And then when we drop those ADP and the P away, it changes shape again and kind of pulls that actin, and that is what causes your muscle fibers to shrink, which ultimately results in a muscle contraction. Metabolic pathways, so a lot of times these things that are happening, processes that are happening in our body might actually be a sequence of smaller reactions, and we might think of it more of just like the beginning and the end result, but there's a lot going on in between. And so this is a case where the products of an earlier reaction can become the products of a later reaction. I'm sorry, oh my gosh, I can't talk today. Products of an earlier reaction become the reactants of a later reaction. And so those are linked together in a product pathway. And so you begin with a particular reactant and then go through several intermediates, right? So A could become B to become C, D, E, F, finally G, right? G is the end product. A is the initial reactant, and these guys are all intermediates in between. And a lot of times, these are being catalyzed by enzymes. We've already talked about those. Enzymes are catalysts, things that help speed up a chemical reaction. These guys bond with the intermediates, which we call substrates, and so they accelerate a specific reaction. And each reaction requires a very specific enzyme. Notice it's E1, E2, E3, right? It's not E1, E1, E1 doing everything. Each of these steps has a different different enzyme to help it out. And so this end product isn't going to form unless all the enzymes in that pathway are there and able to do their job and functional. So a little bit more about how that works. We've talked about how it bonds to the substrate already. And so this happens in the active site of the enzyme. So this is the special spot in the enzyme that is made to link up with the substrate. And when the substrate links up with that active site, it changes shape a little bit. The active site changes shape a little bit, and that little change is going to do whatever to make that reaction happen. So in this case, we've got these two things. It's going to change shape a little bit and force them together, which is going to cause a bond to happen. Or if you're breaking it, it might change shape and force those things apart. We call this the induced fit model. So the enzyme is induced to undergo a slight alteration to achieve optimum fit for the substrates. Okay? So that slight change helps bring them closer together for that reaction to happen. So two different types, degradation. An enzyme can complex with a single substrate, and then it gets broken up into two. Right. So here we've got the single substrate. It connects in with this active site, and then it breaks it apart into these two different products. And notice the enzyme is there and ready to go do its thing again. It doesn't get consumed in that reaction. Versus a synthesis, just the opposite. Two substrates come in, and the enzyme joins them together and releases this new product. And again, the enzyme is still free to go catalyze another reaction. And how this works is enzymes lower what's called the activation energy. And we've talked about this again already. Molecules, they need something to help them react. They're not just going to react on their own. So they usually need some kind of activation, and they also have to come together in the proper orientation. So you have to come together in proper orientation with enough energy. So for example, in this picture, we're trying to make NO2, right? And so if the O and the O hit each other, that's not going to make NO2 and no reaction happens, right? In this case, all right, great, it's making the N and the O are coming together, but that's not the right O. That one's not free to react. So really it's got to get this blue, this nitrogen has to meet with this oxygen to get the NO3 for that to happen. And it has to happen with enough energy for that to take place. And so that's the energy of activation. And this is good, right? Because we don't want these things to spontaneously degrade. We want them to, sometimes there's things we want to stick around in our bodies. We don't want them just to evaporate really quickly. So it's good that we have energy of activation, but sometimes we have to overcome that energy of activation. And that's where the enzymes come in. They lower the energy of activation. So usually they're bringing these substrates into contact with each other, right? And so then they're automatically, putting them in this orientation so that all you need now is the energy to make it happen. Oops. And so this can influence the rate and obviously make it faster. So we often talk about this in energy diagrams, right? Normal reaction would need this much energy, and once you get over this hump, boo, it'll have the reaction will take place. You're left with a net gain, a net loss of free energy, and so this is what you want. And an enzyme lowers that activation energy, and so instead of being from here to here, it's only half as much, so it's easier to get over that hill. So some things that can affect enzyme speed, temperature will generally increase enzyme activity because this is going to cause those molecules to be moving around faster with more energy, so they're more likely to collide with enough energy in the enzyme. It can obviously help that out, so they're more likely to have enough energy. But hot temperatures can also denature or destroy an enzyme. We've learned that in our lab. And so you can kind of see here, like this is enzyme activity, and you can see that there's just definitely this optimum temperature, 45 degrees or so is the best. It'll still work down here, but not as well. Kind of a cool example of this is this mutation that Siamese cats have, where some enzymes are only active at cooler temperatures, and so the enzyme that causes the fur to be darker is at like the tips of the tail and their ears and their face. So this is where they get this darker fur because the enzyme is not active in the white parts of their body. Thought that was kind of cool. Substrate concentration will increase the enzyme activity, which makes sense, right? There's more there, so it's easier for them to find some molecules to put in the right position. And then pH. A lot of enzymes have a particular pH. So if you think of like pepsin, this is an enzyme that's involved in digestion. Digestion happens in the stomach where the pH is really, really low. So that's the happy zone. Versus trypsin, this is not going to be happening in the stomach. It's going to be happening elsewhere. And so its happy zone is more in this basic pH. So cells, luckily, can manipulate this on their own. So they can regulate the presence or absence of an enzyme. Remember, enzymes are proteins, so they can upregulate or downregulate the transcription translation of an enzyme. They can regulate, through that, they can regulate the concentration of the enzyme. And then you can also activate or deactivate enzymes. One of the ways you can do this is with cofactors. So these are molecules that are required to activate the enzyme. And so some of the ones that we'll be talking about later on in the year, FAD, NAD+, NADP+, these are all used, well, these are in cellular respiration. This one's in photosynthesis. Or coenzymes are other parts of the enzyme process can also like be involved in that active site complex to help the substrate bind. Vitamins are examples of coenzymes. And then we can also inhibit enzymes. A common one is a reversible enzyme inhibition. This is something that you would want, where a substance that's known as inhibitor can bind to the enzyme and decrease its activity. So you can have a competitive inhibitor where some other molecule is similar enough in size to the actual substrate, and this competitor molecule could bind to the active site and block the desired substrate from getting in there, and that would obviously slow the reaction down. You can also have a non-competitive inhibition where some molecule binds to a different part of the enzyme, but when the enzyme changes shape, that messes with the active site. And this could be really good. This is a great example of a negative feedback loop, right? Where you've got these guys come together, and that initiates this reaction pathway, and you have this end product. And as the end product builds up, it can then come back and bind in this allosteric site, this other site. And when it binds there, you can see how it's changed the active site, and so now this can no longer accept the initial substrate. So that's a way for the body to like automatically regulate the amount of this getting consumed and end product being produced. And eventually, right, this will get consumed, and that would open up more active sites for more of the reaction to happen. But the bad side of this is when inhibitors cannot be reversible. And so a lot of poisons act this way. So for example, cyanide inhibits enzymes that require enzymes that help produce ATP. Sarin gas is an enzyme that affects neuromuscular junctions, and warfarin is one that's involved in blood clotting. We actually use this, maybe you have somebody in your family who's on warfarin or Coumadin who's had heart troubles. We actually use this to thin people's blood so that they are less risk of like stroke and heart attack. Here's an example of sarin, right? This is the neuromuscular junction. So this is a nerve cell, and this is a muscle cell. And normally, right, these little chemical messengers are being sent across to transmit the message to the muscle cell. We don't want that to keep happening because then your muscle just spazzes. And this enzyme is going to help keep that from happening. It's going to chomp some of these guys down. But sarin comes in and it blocks the enzyme, and so this stuff can just pass freely, and so you end up getting like this overstimulation, and you basically get paralysis because your muscles are constantly contracted. And that's bad because your heart and your diaphragm are muscles, and if those spasm and they can't move, then death results. So the last thing we're going to talk about in this set of notes is oxidation reduction reactions, also called redox reactions. And so these are reactions where electrons are being transferred. And oxidation is when something loses an electron, and reduction is the gain of electron. They are like two sides of the coin. You can't have one without the other. They both always take place at the same time. And one molecule or atom is going to accept an electron that's given up by something else. So a simple example is the production of NaCl, where sodium gets rid of an electron, so it's oxidized, and chlorine is reduced. It accepts the electron. And you think about it, like chlorine then becomes negative, like Cl minus, right? So this charge is reduced. And then in picture forms here, the electron goes from A to B, right? B has been reduced. Its charge goes down. It's gained electrons. and A has been oxidized, has gotten rid of electrons. So this would now be a positive. A good way to remember this is oil rig. Oxidation is loss, reduction is gain. Another one I've heard is Leo says GER. Loss of electrons is oxidation, and GER, G-E-R, is gain electrons is reduction. A couple handy mnemonics to help you out there. So photosynthesis and cellular respiration are two examples of these redox reactions in biology. So these are the overall equations, and you can probably notice that they're pretty similar to each other. Carbon dioxide plus water plus energy gives us glucose and oxygen. That's photosynthesis. And then we flip that around for respiration. Glucose plus oxygen is going to yield carbon dioxide and water, and then it's going to give us some energy. So they're kind of two sides of the same coin. Mitochondria is doing the cellular respiration, photosynthesis doing the, chloroplast doing photosynthesis. So the chloroplasts are capturing the solar energy, and they're going to use that energy to convert the water and carbon dioxide to the carbohydrate, the glucose. The key here is that hydrogen atoms, along with their electrons, are being transferred from water to the carbon dioxide as it forms that glucose. So here we say that carbon dioxide has been reduced. It has gained electrons, right? Because it gained the hydrogen with the electrons, so it's been reduced, and water has been oxidized. It lost its electrons. And the energy required to make this happen is the solar energy. So solar energy is coming in to make the reaction go this way. Just kind of a cool fact, this, if you do this reaction with 1 mole of glucose, this gets us 686 kilocalories. So this is actually what we call a calorie. A kilocalorie so that's how much energy we get from glucose. And then living organisms then can oxidize the glucose in the mitochondria. So the flip side of that, so the mitochondria then, they oxidize that carbohydrate and use that release energy to build the ATP. So cellular respiration, we consume oxygen and we produce carbon dioxide. So it's the exact opposite of photosynthesis. So now the glucose is getting oxidized. It's losing the hydrogen atoms and their electrons. And then the oxygen is gaining those and becoming water. Is gaining those electrons. So it becomes water. And this is a step-by-step process. And then the energy is stored and converted to ATP. And so it's all just a big cycle, right? Carbohydrates that are produced in the chloroplast become fuel for the cellular respiration of mitochondria, and then the mitochondria in our bodies are going to be releasing carbon dioxide, which is becoming a reactant or a substrate during photosynthesis. And so this is basically a redox cycle, where they're feeding each other. And again, these things require each other, right? So the energy doesn't necessarily cycle between the two organelles, though. It flows from the sun, right? So the sun is what's providing all this energy. And again, eventually everything becomes unusable heat when the ATP gets used by the cell. So it's always going to get lost, and we're always increasing our entropy as we go. And that's that one.